Okay, I'll hand over to Paul now to introduce Brian.
Okay, thanks, Hua. Welcome everybody to this joint Biomedical Optics Research Lab CMIC WEISS seminar. I'm delighted to introduce our speaker today, Brian Pogue from Dartmouth. Brian was educated in Canada. He did his first degree at York University, PhD at McMaster University, and then following a time of postdoctoral work at MGH, arrived at Dartmouth, where he's currently Professor of Engineering. I've known of Brian's work really for the best part of 20 years, most notably in optical tomography, particularly applied to breast imaging, in which he was one of the early pioneers of the field. But I've come to realize, perhaps more recently, that Brian's work's really incredibly diverse within biophotonics, and he's made similarly important contributions in a whole range of other areas: photodynamic therapy, fluorescence-guided surgery, and most recently, he's led the way in establishing a new field in the area of in the area of applying optical methods to radiotherapy dosimetry. I think what's also notable is that in all of these areas, and more widely in biophotonics in general, he's been a keen proponent of clinical translation and getting ideas out of the lab into the clinic. He's written a number of really insightful articles on the topic. I recommend those to you if you're interested. And for his own work, he's been active in commercial exploitation by establishing several spin-outs to facilitate this. I guess if that wasn't enough, Brian's also been very active in the broader biomedical optics community, most recently by taking on the editorship of the Journal of Biomedical Optics a couple of years ago. And all of that's been recognized by him being elected fellow of both SBIE and OSA. Okay, so with that, let me not take any more of the time and hand over to Brian.
Okay, thank you, Paul. I appreciate the invite, and it's glad to be here. Can you hear me okay? Just give a nod.
Yeah, okay? I think so.
Okay, I apologize. I decided to do this from home, but my internet seems to be especially flaky today. So very sad, but I hope this works fine. It's certainly much easier than on a plane to London. It's a pleasure to be here talking to you. I've known many good colleagues from UCL over the years and really have an incredible respect for the department there and the education system that you run. So I'll talk about, I chose to talk about translational biomedical optics. That's basically what I do. Talk a little bit about a few things: economics, the science, R&D, and then what I think about publishing a little bit. And again, it's relevant to the editor of BO. I need to disclose I'm involved with companies, Dose Optics and Kel Imaging, both of which I have funded and I'm financially involved with. And then I am reimbursed for being editor-in-chief of the Journal of Biomedical Optics. So when I took over the editor of this journal, JBO, actually went through what I call a personality crisis because I wasn't totally sure who I was. And I think a lot of biomedical engineers fall into this category. What is medical engineering versus, what is being a biomedical optics person? And so I wrote an editorial about it, and I realized you come to, you get a good indication of who you are as a scientist. When you take a hard look at the conferences you go to, you realize who you are as a scientist. And then when you look at the journals that you publish in, you also realize who you are. And so I realized I'm an optics person with imaging science, I would say, and I focus on cancer. And to the students out there, I would just urge you to think hard about these things. What societies do you belong to? What conferences do you go to? What journals do you publish in? You must be involved in 3 of those to be a scientist. And so, you need to find your technological and biomedical expertise areas and dig deep into them. And so,
Sorry, Brian, could I just interrupt you for a second? I think the sound is a little bit broken up, at least to me and a few others. Maybe you could try turning off your video, and maybe that will
I will do that.
Improve the sound.
Is that better?
Yeah, that sounds okay. Yeah.
Okay. I'll keep going, but do interrupt again if it gets choppy. So what is biomedical optics? You know, one of the reasons we don't always recognize biomedical optics is because it's almost everywhere in medicine, all the way from very simple devices to look in the eye or the ear, or more complex devices to do endoscopy, or ultra complex robotic surgery type devices. But it runs an enormous range of technologies. And so it's actually hard to recognize sometimes. And so I just want to start out with a couple of slides recognizing the economic size or footprint of biomedical optics. And actually, just as of I made this slide years ago, but just as of today, this guy's unemployed, so that's quite a nice thing. The impact of biomedical optics is large. And many people, when you say you work in biomedical optics, think of microscopes. And there's certainly a lot of activity in microscopy, but there's actually, and I think I'll try to convince you that there's even more, much more, orders of magnitude more activity in macroscopic optics, taking physiological measurements. So at the microscopic level, you look at cells and very small tissue samples. At the macroscopic level, you look at organ systems and physiology. And, you know, the microscopy world is actually usually very expensive with a small user group, whereas the macroscopic optical world is generally targeted around less expensive technologies, but a much, much larger user base. And, you know, one of the reasons it's hard to wrap your arms around or define biomedical optics is because of this enormous range of technologies. And even if you just think of the cost, you know, you can go from a $10 or $100 otoscope all the way up to a $1 million or $2 or $5 million robotic surgery system. And they're all based on some kind of biomedical optic use within the system. And so this diversity or differentiation in the field makes it hard to fully define. But I did an economic analysis just a few years back, which I published in SPIE Professional, where I looked at just marketing reports of optical technologies in medicine and radiologic technologies in medicine. And it's very interesting that when you add up, you know, ophthalmics, robotics, laser, surgery, microscopy, and endoscopy, you come up with a marketplace that's about $73 billion a year. Whereas you add up all of X-ray, CT, ultrasound, MRI, PET, radiotherapy, it's only about $40 billion per year. So this is surprising to most people in both fields, that the economic footprint of optics in medicine is likely twice the size of radiology. But it's spread through all the different medical specialties. So we rarely recognize it because they just permeate everything in medicine. And then you add in, you know, the obvious growth in mobile and home healthcare products, a good fraction of which are based on the camera or some kind of a light measurement device. And you realize, you know, medical optics is enormous and growing footprint in that. And then just for fun, I, you know, look at the top 20 device companies in medicine, and almost all of them have a basis in biomedical optics as their core technology. And so I conclude that, you know, in terms of technologies in medicine, biomedical optics is the single largest technology sector in all of medicine. And, you know, we don't always recognize that. So I think that's very important to realize, but we don't recognize it because the products are so differentiated. They're, you know, a microscope doesn't look like a home healthcare device. You know, they're all highly differentiated. So that's my little sort of lesson on biomedical optic economics. As we design biomedical optic and I've spent my career designing different types of optical devices. I think it's important to think about the economics of the solution, though, and I'll just show you one little example. Early in my career, I did a lot of work in Colposcopy, which is cervical cancer imaging. And a typical colposcope costs about $3,000. And an advanced colposcope costs about $8,000. And so that's the sort of generic price point, if you will, of what the market can afford. And then I was involved with several companies, and there were probably 5 or 6 different companies that attempted to make very high-end colposcopes. So machine learning, built-in, dynamic analysis, years of development, hundreds of millions of dollars. And most of these devices came out at a price point of about $100,000. And unfortunately, this is very sad, but the field of colposcopy cannot that. No gynecologist can actually afford to buy that device. And so for me, it was a good lesson in realizing that, you can make the most elaborate device in the world, but if people can't afford it, then it will go nowhere and the technology kind of dies. And that's certainly what seems to have happened in this field of hyperspectral imaging for colposcopy. So, design, as you work, again, I think many people here are engineers, as you design a system, have an eye towards the economic medical fit. I mean, you don't have to obsess over it, but you shouldn't be naive to it. And so that leads me to the next section of what I want to talk about is the scientific, because again, I'm not an economist here. This is my hero here. I'm a scientist, I hope. So what's the value of biomedical optics? Well, it's in the electromagnetics, the features like polarization, coherence, molecular interaction, molecular sensing, and the ability that we can create it at the point of care. We need a low-cost device where the patient and a physician are in the same room together when the device is being used. These are the strengths of biomedical optics. And those are not the strengths, for example, of X-ray systems. They're the strengths of So this is my favorite slide. I teach the Physiology for Engineers class. And, in the first chapter, it says that you can categorize the types of molecules in tissue into 4 main buckets, 4 main categories. There are structural things, structural metabolic molecules, there are immunologic molecules, and then there are genetic molecules. And the most important thing for me as an imaging person is that an extreme concentration gradient as you go from the left to the right. Structural molecules exist in the millimolar concentration range. Metabolic molecules like enzymes and respiration molecules, carbohydrates, minerals, exist at the micromolar level. Immunology exists typically at the nanomolar or tens of nanomolar level. And then genetic material like DNA and RNA is way down at the picomolar or subpicomolar level. And so you realize that as you go to the right, your specificity to the tissue increases, but it gets much, much harder to detect because it's very hard to image picomolar concentration of anything, for example. Whereas as you go to the left, it's much easier to detect. So we're really good at imaging structures because they're easy. They're at high concentration. We're much poor at imaging. There's no really way to image genetics without some kind of amplification. Most of the science these days and the active research is in metabolic imaging, immunology. And then as a physicist, this is one of my favorite slides, that recognizing that if you want to image the body, you've got to get radiation through it or in and out so that you can take a picture or reconstruct something. And so I think of the body as a big bag of water. essentially. We're mostly water, and so this is the attenuation spectrum of water across the entire electromagnetic wavelength band. And so if we want to get radiation through the body to take an image, we look for low attenuation. And so there's low attenuation in the X-ray gamma range. We use that for nuclear medicine and X-rays. There is low attenuation in the radio frequency range, and we use that for MRI. And then there's reasonably low attenuation in the optical near infrared range, and we use that for optical imaging. And so you can see just examples of X-ray imaging, MRI imaging, and then optics. And optics does have this this elastic scatter component here, which limits it. But still, we can get light through millimeters to centimeters of tissue, depending on how the device is designed. But again, big picture here is there's 3 windows of how to wavelength bands that we can measure tissue with. And I often kind of joke, if I was going to build a tricorder, it probably would do something like use these 3 different wavelength bands, because they're the ones that can get radiation through tissue. The interesting part about optics, though, is that that's the wavelength band where molecules absorb because of the electronic transition. So a lot of the molecular specific information is in the visible wavelengths. And that's really, again, one of the keys to benefits to biomedical optics. So I work in biomedical optics, and I work mostly at our medical center, although I'm employed by the engineering school. And so I think a lot about how to design a team to make devices. And so here's my friend here again, who says, team, I am the team. And I think a lot of engineers actually fall into this trap of thinking that they can build something and it will be useful, and then they'll just hand it off and it'll be a product. But in fact, successful BME teams have a mixture of expertises. And so not only can we can't just technology push things onto the medical world or industry, and the industry tends to push products into medicine. The medical world thinks of sort of harvesting ideas from academic engineering or industry. But in fact, what's really needed in a biomedical engineering creative way is sort of an embedded model where you've got rapid feedback between academic engineering, medicine, and industry. And so what we try to do is try to get all those pieces together in the same room together weekly. And so my mantra is, if you're not having a weekly meeting with your team, and a very round team, it means that you're not progressing in your project. And so that's what we do. We have created this center for imaging medicine, we call it, within the medical center. We meet weekly with our surgery teams, our radiation therapy teams, our orthopedics teams, and we try to create products that they can use, or test products that they can use, or look for ideas of what they would like us to work on. And this highly iterative, interactive process is the way that we advance biomedical optics technologies. And so, having said all that, I'm going to give you some examples now from our research. And these are examples for new optical systems that can be potentially used to help surgery or radiotherapy improve. And so I'll start out with surgical guidance examples. And so this one is probably familiar to some, which is this idea that we can image scatter of tissue, and we do that with high spatial frequencies. And so here you see a picture of a lumpectomy specimen. And so the surgeon wants to know, is there any cancer on the margin of this sample? And so you can see, actually, we've outlined here, there's a little spot of cancer right there. Everything that's yellow is fat. Everything that's white is some kind of connective tissue. Everything that's red is blood. The fat and the blood are really not interesting. It's about the different types of connective tissue or cancer that's the interesting part. And so it turns out when you image at high spatial frequency, you get a scattered image that comes back. And if you demodulate it at that same spatial frequency, you get a very different scattering pattern. And so you can see here is the different spatial frequencies illuminating it, and then the demodulated image is here. And you actually pull out structural features, the scatter, you couldn't see in the diffuse image, partly because we're making the reflectance more surface weighted, but partly also because we're making it more sensitive to high spatial frequencies. So we have a team of people, a surgeon, pathologist, radiologist, a couple engineers and PhD students, where we're testing this out. And so we take these specimens, we've modified a PerkinElmer IVUS system to do high spatial frequency imaging. This is a system that also does X-ray tomography. So we get an X-ray of each specimen as it comes out. And then here's the reflectance image, and here is the high spatial frequency image. So again, you can see what it does is the high spatial frequency imaging kind of negates all the fat and blood content, and you really pull out what I would say is the connective tissue or the tumor-associated collagen. And just to show you some examples, this has been a fascinating trial. So here's what the specimen looks like. These are all specimens that have intralobular carcinoma in them, and so that's a very interesting type of breast cancer because it's kind of microinvasive and hard to distinguish. But here's what it looks like in room light. The high spatial frequency scatter image is here, and then the X-ray image is here. And what we've shown, and we're just about to publish this, is it's actually fascinating that the high spatial frequency image, because it pulls out collagen structures, looks a lot like the X-ray CT. And so, but it's just at the surface. So it's kind of like a surface scan of X-rays, if you will. And so we present this to the surgeon, and we're doing a clinical trial now to determine if that's useful to help guide the resection. And can they see residual cancer left on the edge of the margin? So we reveal tissue structures, the presentations in real time during surgery, and we're kind of right in the middle of our ongoing clinical trial with this technology. We just published a sort of advancement to this where we realized that high spatial frequency is one way to do it. There's actually an even easier technology, which is active line scanning. So you just take a line of light and scan it. And as long as you just pull out the reflectivity at that very narrow band, it's kind of like a confocal microscope, but it's wide field. And you get extremely high dynamic range. And so for subtle scattering differences. And so we published a paper just this past year in December on narrowband line scanning for scatter microstructure. So we're interested in that. But, to me, the more exciting part would be to get to guiding surgery with molecular information. So I've been a number of years trying to push us towards getting more insightful by molecular guided surgery. And so actually I'm writing a review paper on this now. If you think about the history of fluorescence in medicine, it actually goes back to the 1960s, where fluorescein angiography was developed for looking at the back of the eye. And so that was approved for commercial use back in the 1960s, as was indocyanine green in the 1990s. Indocyanine green has subsequently become even more useful for open surgery applications like tissue perfusion imaging. So you inject this contrast agent, and then the fluorescence image can show you, where there's blood flow and perfusion in the tissue. It's used both in open surgery and in laparoscopy. And then just in the past decade or so, I would say there's been an exciting evolution of using molecular contrast agents. ALA, or aminolevulinolic acid, which produces protoporphyrin 9, has been shown in neurosurgery and in cystoscopy. And so this is, to me, this is the milestone marker of the next generation of molecules, because it's a molecule that is produced in the mitochondria through the heme synthesis pathway. And so it's an indicator of not of just structure, but of metabolism of the tissue. These are growing markets. So I did a survey in clinicaltrials.gov, which is the American website listing all clinical trials that are registered. And there are close to 1,000 clinical trials on some kind of fluorescence in surgery. You know, about a third of them are non-specific, I would say. About a third are metabolites. Actually, that's not true. About 2/3 are non-specific fluorophores. About a smaller number are metabolites, and then a relatively small number are immunologic probes. But having said that, you know, 1,000 clinical trials is pretty exciting. It means that there's a lot of activity in the field. And again, if I go back to my physiology of molecules slide, most of the action, most of the interest is in trying to image tissue metabolism or tissue immunology, because those are more specific than things like just vascular perfusion or connective tissue. So we got into this. We got a large grant funded from the National Cancer Institute to try molecular imaging with anti-EGFR, epidermal growth factor receptor, with an AF molecule. It's a very small 56 amino acid peptide that we bound with a fluorophore. We produced it for human use, and then we are in the tail end of 3 clinical trials with this now. And this has been pretty exciting because it's an immunologic probe that labels EGFR positive tumors and allows the surgeon to see that as they're guiding the resection. So there's a lot of nuance to this field, but it's progressing pretty well. When we got into this about a decade ago, people were still kind of building their own fluorescence-guided surgery systems, and it was an interesting time. And in 2016, we published a review paper looking at the different systems and sort of coming up with our top 6 list of the features we would like to see. Things like real-time overlay of white light and fluorescence, being able to work in ambient room lights, good ergonomics, so forth. There was a good study. I would say now, though, that this field has matured, and this is very exciting, to where there's now 6 or 7 different FDA-approved systems. And so the field has now, I would say, transitioned to more of a procedure-based research field. But that's exciting because it means that we are progressing in the field of, you know, guiding surgery by molecules instead of just structures. So I'm going to leave that there because, again, that's more procedure-based research now. And on the device side, I got involved in radiation therapy work, again, about a decade ago, where we realized that we could image the Cherenkov light coming from patients when they're getting radiation therapy. And so this has been an exciting odyssey that goes all the way from sort of idea to commercialization and product development. And so you can see some examples here of Cherenkov coming from tissue. So radiation therapy is a field of treatment of cancer using particle beams. And so an electron beam is excited through this linear accelerator, hits a target, and then a very high energy X-ray beam comes out. And so these linear accelerators, or LINACs, are very highly developed. They use multi-leaf collimators to shape the beam. That beam is targeted to each patient's cancer tumor. And so a treatment plan is developed for every patient, a custom treatment plan. And so here you can see a modulated plan where the shape is being altered as it treats different parts of the tumor. And so this is a cartoon of a prostate treatment. And again, the beam, and cartoon-wise, is going through the patient this way, then going through the patient this way. And what they do is a dose buildup in the target volume. And they try to minimize the dose buildup in the surrounding normal tissues so that you get a enhanced treatment in the tumor and try not to damage the normal tissue as much as possible. But this is a cartoon, right? So X-ray beam delivery isn't visible. You know, we've all had X-ray systems, X-rays and dental X-rays, for example. You can't see the beam, but you can see the dose delivery through this very, very tiny light signal called Cherenkov light. And so the Cherenkov light is familiar to most of us. If you see pictures of used nuclear fuel bundles that glow blue, that's Cherenkov light. You can see that same blue glow in a patient when you hit it with X-rays. The interesting thing, though, in tissue, like most things with light, biomedical optics, is that the blue wavelengths are absorbed. And so Cherenkov is actually broadband, and the light wavelengths that survive are in the red and near red. So we capture light signals from about 600 to about 950 nanometers, and that's the Cherenkov light we see coming from human tissue. And so we spun off a company, and so I'm involved with the company financially. It's a great team, though. We developed a custom camera system that would time gate to the linear accelerator pulses and capture these very small Cherenkov light signals just in the 4 nanosecond bursts of each linear accelerator pulse. And so the cameras are designed around an image intensified, a time gated image intensifier, which couples through a relay lens to a camera. And then very importantly, that camera is controlled and fed into an FPGA board, which does a lot of image processing on the back end. And then the USB 3 is used for the image out as well as power in. And so we capture real-time video treatment of the, by capturing these Cherenkov light signals that come off the tissue. The fascinating part of it is actually we're imaging at the single photon level. So we're actually getting, the image we report is made of actually relatively few photons. We kind of time average over several pulses because otherwise it's very spotty. But you can capture single photon level images with the room lights on, with the camera up to a couple meters away from the patient. And so here's a little picture of our TrueBeam system in our radiation therapy department. The cameras are mounted in the ceiling. There's a lot of monitoring equipment in these rooms. So the cameras are just one more piece of monitoring equipment to capture the radiotherapy treatment. So we published a paper just last year showing in the stereotactic treatment. This is a patient who is getting a treatment to the back of their eye. There's a tumor at the back of the eye. And so you can see they're awake, they're breathing, blinking, and this is actually the beam walking through the patient's head as it arcs around. And so we were able to capture the beam in real time. And actually the point of this paper was we could even see the Cherenkov light coming out of the pupil of the patient. And so we knew that the beam had actually crossed into the orbit of the eye at a certain part of the treatment, and which was not supposed to happen. So there was an interesting sort of side observation. But most importantly, what the beam does is the Cherenkov imaging does is it allows a real-time display of the beam as it hits the patient's tissue. So you can see a typical breast treatment here where the beam is delivered from 2 different angles, and the software displays that in real time, and so that you can, the therapists who are doing the treatment can see the treatment in real time. Here's a patient getting whole breast radiotherapy. And again, you can see it's a real-time video. Some treatments where the shape of the beam does change, and so we can capture that and display it. And so we just a few weeks ago now published this paper in our clinical experience from Leslie Jarvis, who's our the radiation oncologist doing most of this work. We surveyed 65 treatments, I think it was, and found 6 cases where things happened that probably shouldn't have happened, where there was radiation a little high on the neck in one case. Not catastrophic, it's just it shouldn't have been that as high as it was. sometimes the patients lower their head and they get a little too much dose on their chin. There was one case where the bolus wasn't placed properly, so this place it was. And then in this day 2 here, the bolus was a little lower, and that's a change in the dose at the midline. Here's a couple cases, one where the dose was a little too low on the armpit, one again where it's a little too high on the chin, the neck, and then 2 cases. Oh, this is the same patient. It's a case where the patient couldn't keep their head straight, and so they were getting extra dose on their chin. And then that's the bolus one. Yeah, so we're doing some radiotherapy work, looking at head and neck patients as well, tracking the Cherenkov light from daily fractions to see if there's value in just determining when to replan head and neck patients. Rachel Hatchadorian had a great paper this past year where she showed that we see differences in Cherenkov intensity because of the tissue optical properties, which is not surprising to most biomedical optics people, but in a dense breast, less light comes out than in a fatty breast. And so what she showed in this paper was that there was a direct linear correlation between Cherenkov emission decreasing as CT number increased, because CT number is essentially a surrogate for blood volume. And so as the blood volume increases, the light output decreases. And so she showed that you could correct for this and use the CT as sort of a calibration factor for attenuation and calibrate the Cherenkov to be absolute dose or closer to absolute dose. So that was a fun study. So that's the end of my radiotherapy. I'm just going to show one more little vignette before I talk about publishing. We have been working in molecular imaging and radiation therapy. And again, this is one of my obsessions, I would say, is this idea of, can't we guide medicine by molecular information instead of just structures? Well, when you realize that Cherenkov light is produced from radiation, when you put an X-ray beam through tissue, you're producing Cherenkov light through the volume of tissue. And so you could excite molecular reporters inside the tissue, and that luminescence from them would be picked up by our time-gated camera. And so we advanced this with the idea of imaging oxygen in tissue. And so we did some rat studies where we put PO2 sensors in lymph nodes, individual lymph nodes of rats, and showed that we could image the PO2 inside a single lymph node with an X-ray beam excitation. And that was published back in 2016. We showed, that was this idea that you could do high resolution molecular sensing if you localized the probe where you wanted it to be. So that was kind of a proof of concept. And then subsequently, we've continued this. And this is all in collaboration with Sergei Vinogradov at the University of Pennsylvania, who designs these really elegant molecules where there's a phosphor in the middle. And then it's surrounded by a dendrimer shell that controls the oxygen diffusion. And then it's pegylated out on the exterior so that it's biocompatible. And so he calls these oxy-4 molecules, and they absorb the Cherenkov light in the visible. They emit in the near infrared at 700 to 900 nanometers. And their lifetime, most importantly, their lifetime is a unique indicator of the local PO2 or oxygen. And so we showed this paper a couple of years ago that we could do high resolution oxygen sensing of tumors by reconstructing the PO2 in the location of where the emission came from. And so we were able to image PO2 in these tumors with 100 micron spatial resolution. So that was really exciting for us, an illustration that you could do light sheet macroscopy with radiation beams. We kind of swept radiation beams across these tumors. And I think of it's sort of like light sheet microscopy, where you put a sheet of light through a sample and then allows you to image thicker samples. What we're doing is light sheet macroscopy. It's imaging through whole animals with radiation beams. And so we've done a bunch of work looking at PO2 histograms now and showed that we could get high resolution histogram information about the oxygen in tumors during radiation therapy. And I think I'm just going to skip through this stuff. But basically, we could track tumors through the course of radiotherapy and follow their PO2 histograms. Again, oxygen is highly heterogeneous in cancer tumors, and so the histogram is actually what you really need to report instead of just the average PO2. But the histogram, the changes in it, tell you a lot about how radiosensitive that tumor will be. So that's, I just showed you 4 examples, and just that's kind of to wrap this up is, I showed you surgical imaging with scatter, surgical imaging with molecular contrast agents, radiotherapy imaging by Cherenkov, and then PO2 sensing in radiotherapy with a luminescence capture. So those are, the 4 application areas I spend a lot of my time in. But I want to end just with kind of a statement, and I think we've got a couple minutes here, a statement about publishing. And I was really, I've done a lot of thinking about, what I consider success in academics, and is publishing a good paper the end? When getting the next grant, is that the end product? If you go back to 2016, there was actually a whole series of papers in Nature where they did a survey of their readers, and they said, do you believe that there's a reproducibility crisis? Meaning that, do you believe that what's published is actually true? And they showed that actually the majority of people believe that there was a crisis. In fact, it was up to 90%, I think, believed that there was either a slight crisis or a major crisis. And they talked about what are the factors that contribute to issues around reproducibility. But basically that, as we get pushed into publishing high impact papers, there's a greater and greater chance that it's not really a reproducible observation that's being published. So 90% of researchers say there was a crisis. And so this actually led to a large number of think tanks, interest groups, government panels about how to make biomedical research more honest, or how to make sure that we don't distort things as we go to publish them. Are we producing productive, or another way to say it is, are we producing productive science where we can believe what's published in papers? And so this led me to being interested in this idea of revising how we reward scientists. And this, one of my favorite papers was this one in 2014, where they advocated for the PQRST approach to reward science. And P stood for productivity, meaning, so you measure productivity or success by productivity, grants and publications, that's fine. But then what you also need to care about is the quality. Do other people cite it? Do the techniques used follow quality standards? And so there's a lot of emphasis these days on proving that your research used quality tumor samples, for example, followed quality procedures. But then they go further and say, well, is it reproduced? Can somebody else do that technique or see that result? Does that research group share its data, or do they provide access to the resources or data that they produced so that other people can try it? And then T stands for translation, meaning did that lead to some kind of a clinical trial, startup company, licensing of the technology? And it's not really until you look at this whole range from P, Q, R, S, T that you realize that the science was true science and reproducible. And so, I would argue that actually when we look at scientists and academics now and from promotions and tenure, we largely just look at productivity and quality. And we sort of draw a line, and we don't really think about reproducibility, sharing, or translation a whole lot. But there's a renewed emphasis, I would say, especially in engineering schools, around some of these latter things as independent measures of success. And so that's led me to be more interested and active in spinning off technologies into companies. And so I've been involved in these 2 startup companies, one doing Cherenkov imaging, one making fluorescence surgery devices like phantoms and cell phone-based diagnostics, just because I want to see the things that we invent get turned into reality and prove that they work. And so that translation, I think, is an important piece of biomedical engineering today. And so as we think about the definition of success in publishing, and so I wrote an editorial about this, but basically we need to rebalance things a little bit less towards publications grants and a little bit more towards reproducibility, sharing of things like data, models, and code, and then seeing that things lead to a realizable translation in terms of either clinical trials or startup companies. So that's the end of my talk. I'll turn on my video, see if this works or not. I hope you can still hear me. Can people? Yeah,
yeah, we got all of that.
Good. Okay.
Thank you. Thank you very much, Brian. That was excellent and very thought-provoking topic to finish on there. Are you okay to be around for a few minutes to take questions?
Sure. Yeah. Yeah, I'm here. Yeah.
We're coming up to our allotted time, but perhaps we could carry on for a little bit if you've got time, if we need to.
Sure.
Good.
I know nobody asks questions on the Zoom forum anymore, but if there are any, I'm happy to answer.
Yeah, I didn't see any in the chat. So yeah, please can you raise your hand if you'd like to speak.
I'll just stop.
Okay, I don't know if that's, yeah. I can't actually see everybody. Hua, do you know if anybody's put their hand up? If not, I'll.
I don't think so.
Okay. Okay, Brian, perhaps I could ask you a question. On these high spatial frequency imaging, you showed some really high resolution images. Can you just elaborate a bit on the instrumentation that you used, and particularly how you illuminate the tissue?
Sure. Yeah, it's actually incredibly simple, really. We, the tissue is put into a glass or plexiglass case, so it's flat on the top and flat on the bottom. And in this case, it was just a projector was projected with R, G, and B, red, green, and blue colors, and we captured that with a color camera and then demodulated at high spatial frequency. And just like Modulim, the company, we use 3 different phases of the light spatially, sort of, and you square each of those, and then that demodulates the image. And so you get a high, we actually don't use low spatial frequency at all. We just use the highest spatial frequency, which I believe is 1 inverse millimeter. And this is imaging very large samples, they're as big as your hand. So, 10 centimeters across. A spatial resolution of 1 millimeter is pretty good, actually, because we're doing macroscopic imaging.
So you get the high spatial frequencies by filtering the received light, or?
So, okay, so the intensity is a sinusoidal intensity, right? So it's displayed onto the tissue. We image that reflectivity, and then we shift the phase 3 times. And so we demodulate those images at that spatial frequency. And so we're not looking at diffuse light anymore. We're looking at, we're looking at reflected light at the high spatial frequency.
Okay, so you vary the illumination pattern.
Yes.
And demodulate.
But for the most part, we just use the highest spatial frequency. We don't really use the lower frequencies.
Question from Ilyas.
Hi, Brian. Can you hear me?
Yep.
Yep. Yeah. Excellent. Yes. So good to see you again, and an excellent talk. So I just got just a quick question. So I was interested, on some of your latest results that you found that the intensity, of course, you receive, it would depend on the tissue and, and what is in the tissue, like the increased blood volume will reduce, your reflectance, that's kind of expected. But I was wondering, can you use that as a biomarker of some sort? So can you, for example, try to get, a quantifiable information about blood volume, perhaps, or fat lipid content through that?
Oh, you're talking about the Cherenkov light, or?
Yes. Yeah, sorry, yes, about the Cherenkov, yeah.
Yeah, the Cherenkov light. These are patients who are getting radiation therapy, right? And so most of the question and concern is the treatment correct? Is the dose being delivered in the right spot? And is the dose the right amount of dose? So we focused our energy around the idea of correcting for Cherenkov intensity variations, because as blood absorbs, you decrease the Cherenkov. We think that by calibrating with the CT number that we can correct for fibroglandular versus fat content in the breast. There's also some activity in using the blood vessels. I don't know if you saw, but you actually can see the superficial blood vessels of the breast as sort of fingerprints or inherent fiducials, if you will, to verify that the treatment was in the same spot every day. That's really interesting. And we have a student working on that. But that, I think, could be very useful for automated verification of delivery. And as you may know, I mean, in radiation therapy, things are just progressing towards automation. I mean, I would not be surprised in 10, 20 years if, the number of people in rad-onc are much, much fewer, because the whole process is almost completely computer automated. So I can imagine this type of imaging being used for something like that.
I mean, it'll be interesting, as I said, whether you can use some, from the Cherenkov emission to do some sort of a spectroscopy. That's what I was wondering, if that's if that's sort of a possibility, and then you can use that and feedback, to your clinicians about, what effectively is happening, or what type of tissues, they're sort of injecting radiation therapy into.
Potentially oxygen saturation, for example. I mean, we have to be a little bit careful though, because we know the information is only coming from less than a centimeter of tissue. It's not deep. It's not Cherenkov from inside the breast or tissue. It's just in the very top layer that we're imaging. So we don't want to overstate what we can do, but potentially things like oxygen saturation or blood volume can be quantified.
I have also just one last question, and it's related to the last part of your talk, which I find very interesting and very sort of thought-provoking as well. Do you think the sort of the translation part, like so the spin-out company part, contradicts the sharing of information part?
Yeah, well, it's a great question. That as soon as you get into the details of like sharing versus technology spin-off, obviously they're competing with each other, right? But I think every investigator, and this, every investigator has to make a decision, right? Am I going to spend my life advancing this technology in a startup company? If the answer is no, then you fall more on the science side. And then, and there's projects that I work on where I'm really much more of a scientist, right? And I would just like to share the data with everybody and anybody. And we do that. And I think that's important. And as the journals more and more are pushing them. You can't publish in Nature or Science journals without sharing your data. So I think that's the right way to go. If you want to publish a paper in a good journal, you should be complied or obligated to share your code and your data. I think that's the way we're going to advance science. And JBO has adopted those policies, although the policing of it is another question.
Absolutely. Thanks for the comment. Thanks. Cheers.
Yep.
Okay.
Thanks.
Jamie, I think you were next.
Thanks, Paul. Hi, Brian.
Hey,
Very nice, inspiring talk. I think what you just said probably started to answer this. On the reproducibility question. Do you think there's a reproducibility crisis in biomedical optics? And if so, would you comment on its nature?
Yeah.
And what do you think needs to happen to increase reproducibility of the work that we do?
Yeah. I'm a doubter by nature. Like I just tell my students, I read a paper and the first thing that comes out of my mouth is, I don't believe that.
Yeah.
Which is a terrible thing, but that's just who I am. So I think we all know that sometimes things get published that are questionable. I'm not sure there's a crisis really. I don't publish in Nature and Science that much. It's more cell biologists and sort of genetics people who publish in those journals. I might publish once in a while, but the pressure the pressure to publish in those journals is intense, right? And it makes a difference in people's careers and lives. And so that pressure has created a situation where scientists are pressured to maybe publish things that aren't as fully verified as they could be. So I don't think it's that bad in biomedical optics. But I think where I see it happen in biomedical optics is, what's the signal to noise of a system, right? And is there really enough signal to noise to be useful? There's a lot of papers published where I think the answer is probably no, but yet we publish it anyway because we think, well, with technological improvements, things could get better. So that's an issue. But to get to your last question, how do we make it better? I don't think it's critically bad, but I do believe in sharing data, sharing code, telling people what you're doing as openly as possible. And then if something truly works, I think that engineering academics have an obligation to get involved in spinning it off. And so, for example, now, I'm not interested in writing a patent unless we're going to spin a company off. And I'm going to take a fraction of my life and devote it to seeing that technology get spun into a company.
Right. So the point is, if it doesn't work, then that won't happen. So it really has to work. It really has to be reproducible.
Yeah. So it's a long, complicated topic, but I think those are some simple ideas. Yeah.
Okay. Thanks, Brian. Thanks again. Really enjoyed the talk.
Good to see you.
Brian, just on the reproducibility issue, in other fields, in some journals, they're publishing reproducibility studies. Do you see any move to doing that in biomedical optics?
Yeah, that's an interesting and tricky question. People don't typically get any credit at all for reproducing other people's work, right? In fact, that's like a common critique of reviewers is, so this is just reproducing what so-and-so did. But I would say this, if the observation is really, really important, then obviously it needs to be tested and reproduced, or really, really controversial, it needs to be tested and reproduced. And sometimes, sometimes scientific communities galvanize around that thing. I don't think every study needs that, but I think important ones do.
Yeah, it does feel like it needs the community to want to do it and see the need to do it.
Yeah,
really
but I also believe in the free market system, right? Which is, if it's true, and it's useful, the free market will bear it out, right? Companies will produce a device and it'll sell, presumably, if there's actually value in it. So
it'll come out eventually.
Yeah.
There's a question in the chat from Dale Waterhouse. Hi, Brian, very informative talk. How do you balance perfecting technologies in the lab versus pushing towards clinical translation? And how do approaches differ between academia and industry? Does this cause conflict?
Yeah, that's a great question. Every academic struggles with that. And I think Dave has a paper on this that's published in Nature BME, Dale. So, the challenge is, you make it as perfect as you can until you run out of money, and then you do the clinical trial, right? Academics and industry do that. Even industry doesn't make perfect devices. They make it as good as they can within the financial constraints of their system. And then they do a clinical trial, and sometimes it fails because it wasn't designed as well as it should have been, which is unfortunate. But you have to make those difficult choices. Yeah, I mean, economics, impacts engineering, right? And there are many devices we could make that are just too expensive to be valuable to be used. So it's a tricky topic.
Okay,
in terms of
industry. Sorry.
Go ahead. Go ahead.
Okay, I just saw one other question, or somebody put their hand up earlier. Hubin, did you want to say anything? Okay, looks like his hand has gone down. Good. Okay. All right. It looks like we've no more questions. So I'd just like to thank you again, Brian, on behalf of everybody, for a really excellent talk, and thank you for your time and contributing your thoughts.
Well, thank you. I appreciate how many smart people are online here, and I'm awed and that they've taken time to listen. But thank you for the invite. It's really a pleasure. Not quite as much fun as going to London, but
yeah.
very close.
Hopefully soon, one day.
Thank you.
All right. Thanks again, Brian.
Okay. All right. Sure. Cheers.